Namaskar. Today we will study the negative regulators of cell cycle. Before that, let us see what is the cell cycle. In the cell cycle, actually, what is the cell cycle? Cell cycle is the stages. It consists of the stages through which a cell passes from one division to the next division, and it may be consist of interphase and M phase. Interphase consists of G1 phase, pre-DNA synthesis, S phase, DNA synthesis, G2 phase, post-DNA synthesis or preparation for or mitosis. And after the cell division, the cell may undergo the resting phase G0, and the positive regulators of cell cycle, they help in progression of the cell from the G0 to G1, G1 to S, S to G2, G2 to M, like that. And the negative regulators of cell cycle act on the checkpoints. There are two checkpoints, one before the DNA synthesis and two before the mitosis. So the negative regulators of cell cycle, mainly p53 and retinoblastoma RB gene, they act in these checkpoints. And in addition, there are CDK inhibitory proteins, CIP, and inhibitor of kinases, which can act as the negative regulators of cell cycle. So today we will discuss mainly about the negative regulators of cell cycle. As we know, the cyclin and cyclin dependent kinase CDK form complex because the CDK is inactive in itself, and when it binds to cyclin, they form the complex cyclin CDK complex. And there are different cyclins and different CDKs, and those cyclin and CDK, they form complexes, and those different complexes, they help in transition from G1 to S, regulation of S phase, then from G2 to M phase. So different cyclins and different CDK4 complexes, they regulate all these phases. And those complexes are inhibited by specific CDK inhibitory proteins. So these p27, p16, p21, they are also negative regulators of cell cycle. So when these negative regulators will act when there is excessive cell division or when there is DNA damage, in both the cases, the negative regulators start working. So when there is DNA damage, let us first consider when there is DNA damage, immediately there will be recruitment and activation of various protein kinases. We call them as ATM or ATR kinase, and these are members of phosphatidylinosyl 3 kinase family. Then these ATM or ATR kinase, they will recruit and activate additional protein kinases called CHK1 and CHK2 kinase, and these kinases, they release the MDM2 from p53 MDM2 complex. Usually the p53, the negative regulatory gene, is in the inactive form because of the complexion with MDM2. So the p53 MDM2 complex is the inactive form. The p53 becomes inactive by forming a complex with MDM2, and these CHK1 and CHK2 kinases, they release the MDM2 and phosphorylate the p53, so there will be active p53. And actually MDM2, by forming complex with p53, helps in the degradation of p53. So the negative regulator is neutralized there by MDM2. But when the MDM2 is released by these protein kinases activated by DNA damage, they keep the p53 in the active form, and this p53 regulates the transcription of certain CDK inhibitory proteins like p21, which will inhibit the cyclin E CDK2 complex. So p21, it inhibits the cyclin E CDK2 complex, and p21 also inhibits cyclin B CDK1 complex. So cyclin E and CDK2 regulate the transition from G1 to S. Cyclin B CDK regulate the progression from G2 to M. So here we can see here G2 to M cyclin B CDK1, and here G1 to S cyclin E CDK2, and so both these will be arrested by this P21, and P21 transcription is regulated by P53. So P53 causes the transcription of P21, which will inhibit the cyclin E CDK2 complex at the checkpoint 1 and arrest the cell in G1 phase, and also inhibits the cyclin B CDK1 complex at the checkpoint 2 and arrest the G2 phase. In this way, the cell division stops and the cell undergoes apoptosis, and in this way P53 acts as the negative regulator of cell cycle. There is another mechanism by which P53 acts when there is excessive cell division. So in case of excessive cell division, there will be excessive MYC production, which will activate another protein called ARF, and this ARF will form complex with MDM2, so P53 will be released and P53 becomes active and can regulate the transcription of P21. So in this way, P53 acts as the negative regulator of cell cycle. And the second one, the retinoblastoma gene, how this retinoblastoma gene acts. Let us see, when there is excessive cell division, the cell division is induced by mitogen, and the mitogen receptor is a tyrosine kinase linked receptor, and when mitogen binds to the mitogen receptor, then it will follow the RAS-RAF pathway. First there will be GTP-GTP exchange in the RAS, so then it will phosphorylate the RAF will phosphorylate the MEK will phosphorylate the MAP kinase activate the transcription of regulatory protein MYC and activate the G1 CDK like cyclin D CDK4 complex, and this complex phosphorylates the RB retinoblastoma gene, and retinoblastoma gene acts as a negative regulator because it inactivates the transcription factor E2F. So E2F protein is released, and this E2F protein activates the G1 S-cyclin like cyclin E CDK2, cyclin A CDK2, and help in DNA synthesis, regulate the S-phase genes and causes cell division. Cyclin E CDK2 also again phosphorylates the retinoblastoma gene and releases E2F protein. So in this way, the cell division is activated by the cyclin and CDK complexes, which is inhibited by retinoblastoma because retinoblastoma gene inhibits the E2F, the regulatory transcription factor. So retinoblastoma protein actually inhibits the regulatory transcription factor, and this regulatory transcription factor causes the S-phase gene transcription, which helps in synthesis of DNA replication enzymes. Hence there will be DNA synthesis and cell division. And the positive regulators like cyclin CDK complex, cyclin D CDK4 complex causes the phosphorylation of retinoblastoma, so there will be release of E2F and there can be this action. And retinoblastoma protein or gene, by inhibiting E2F, acts as a negative regulator of cell cycle. So this is all about the negative regulators of cell cycle.